Okay, so we saw what happened to inside the cell for the cells to divide from one cell to two. Okay, so today we're going to go through chapter 9 and chapter 10. And the chapter 9 title is the Links in Life's Chain, which means we are looking at into genetics from today. So the question is how we look like our parents. Okay. Before that, we have to understand how the cells to divide. In other words, how you can make two cells out of one cell. Okay. You have to grow, and the cell has to increase their numbers, or as in the movie, the soccer player get injured, you have to heal, the new cells has to produce. So from the starting one cell, you have to divide into two cells. Okay? So the question is how we can make two cells out of one cell. Okay, you can make it bigger and just divide. That's easy. But the important thing is the genetic information inside the cell. Okay? So the key point is you have to make exact copy of DNA information and then give it to the new cell. Okay, that's the key point of chapter 9. Okay, it's called mitosis. Okay, chapter 10 we'll talk about a little bit different question. How are we going to prepare for the next generation? Okay, Veronica has two last names, right? Your father's name and your mother's last name, right? So in Latin America, you pick last name from mother, last name from your father. If you keep continuing, what happened? Your grandchildren will have what? 8 names? 8 last names? No, 4. You have to drop it, right? Right. So in other words, in order to prepare next generation, what do you have to do? You have to give only half of what you have. Then the other half will come from mother or father of the child. Okay, that's chapter 10. It's called meiosis. Okay, so we'll go through first mitosis. Okay, so an introduction to genetics. So now this will be basic understanding of how we look like our parents, and then our children will look like us. Okay, we all know now that DNA is the information-bearing molecule. Okay, DNA has the information. We'll go through what the information is. Okay, what is this information is used for? To making RNA, and from RNA to protein. Okay, protein is showing what we are. Your hair color, your shape, your muscle, all the things are expressed by proteins. Okay, so the DNA contains the information how to make proteins. Okay, so what is the information inside the DNA? We know DNA can have 4 different information. Okay, GATC, whatever the order you want to remember. Adenine, thymine, guanine, cytosine. You don't have to even remember the whole name. Just remember ATGC. 4 different base is the menu in DNA. So with this 4 different, you have to create the information. That's DNA language. Okay, GGG, CCC, whatever the sequence is the information. Okay, so then this is how you store this information in DNA. We learned that usually DNA inside the cell has this structure called, what structure is it? It's called double helix. Okay, consists of 2 strands, anti-parallel. One strand goes this way, the other strand goes that way, and it's intertwined. Okay, it's called double helix. Okay, the outside handrail. Okay, you can be compared to stairs. Okay, if you go to a battleship or if you go to lighthouse, very narrow space, they have to put stairs. Okay, so that handrail is made of phosphate and sugar. Do you remember the three components in DNA? Base, sugar, phosphate. Okay, there are two. Phosphate and sugar forms this handrail. Okay, the base in the middle is the step, okay, upstairs. Okay, so that has information. AG, I don't read here, AAG, whatever AG. This is the information DNA stores. Okay, remember it consists of two strands. Okay, and it's called complementary. Means if there is A, there is always T comes as a pair. Okay, if G comes, there always is C. Okay, G, C, A, T is pairing. Okay, so that's the information that's stored in DNA. Okay, so the reason why Watson and Crick got the Nobel Prize, as soon as they come up with the model, DNA should look like this. They come up with a correct structure model. Okay, as soon as the structure comes out, people start to understand how they can copy the exact information to the next generation. How? Because it's complementary. If you open up this one and then use one strand as a template, you just fill in the information. Because if there is A, there should be T, right? If there is a G, it should be C. It's automatic, almost automatic. That means complementary. Okay. So go through. Yeah. Then what is the gene? Okay, gene. Okay. So each series of protein specifying base, in other words, from here to here in DNA, G, G, A, T, C, C, you go a long way. And one unit, and that's information, is making one protein. And that's one unit, which we call it a gene. Okay? So a gene, a DNA information, is copied to RNA information. That information will be used to make one protein. That's the definition of gene. Okay? And what about genome? Well, before we go, so how this DNA information is becoming a protein? Okay? So information in the sequence of DNA is exactly copied first into RNA molecules called messenger RNA. Okay, this copy is temporary one. You can use it, you can get rid of it, but DNA is the stored information. Okay, so messenger RNA coming out of nucleus, because we keep the DNA inside the nucleus, right? Coming out of as a RNA molecule. Where do they go? They go to the place called ribosome. That's where they make protein. Okay, so what is the language of protein? DNA language is GATC, right? What is the language of protein? Loud? RNA? No. Amino acid. Yes, they have 20 different amino acids. They can choose 20 different amino acids, and that will make a specific protein. Okay? So DNA and RNA use very, very similar language, right? GATC, RNA language is GAUC, right? But protein use totally different language, amino acids. Okay? So what do you have to do? Ribosome has to translate it from one language to another language. That we call it translation. Okay? So they will make protein. So what's this, the whole thing? From DNA information become RNA, RNA become protein. We call it gene expression. The information stored in gene, in DNA, will be showed up as an appearance of your hair color, shape, muscle, whatever, body size, height, weight. Okay, this will be showing up from the DNA. Okay, that's what we call it gene expression, or that's how to make protein. Okay? All right, this slide summarizes the whole thing. Okay? So we have this genetic information, okay, how we look like, or, you know. And that stored information is in DNA in the nucleus. Okay? That information is transcribed, it's copied, okay, to RNA molecule. In this case, it's called mRNA, stands for messenger RNA. They'll travel out the nucleus. Where do they go? In this case, ribosome in cytosol. There was another case, the ribosome can sit on endoplasmic reticulum. Right? There are two places you can make protein. Okay? The other one is RER, rough endoplasmic reticulum. Those proteins we will ship out. But this one in the cytoplasm will use them. Okay? So they go to ribosome. Ribosome will translate the language of RNA into language of protein. Okay? We'll come back to that point. All right, so next word is called genome. In Korean, it's a little bit bad word, genome. Okay? It's a bad word. This is how we pronounce it, okay? So genome is the whole gene set. All the DNA information that you have inside the cell. It's called genome, okay? We know all the DNA is stored in nucleus, right? But there are some small amount of DNA inside the cell, not in nucleus. Where? Anybody? All the DNA should be stored in the nucleus inside the cell, right? Okay, but there are exceptions, two exceptions. In some cells, has separate small DNA. Where? It used to be microorganism, and they decided to come into the cell. Mitochondria and chloroplast have their own DNA, small DNA. But don't worry about it. Just let's say genome means all the collection of the genes, all the DNA information that you have. Okay? The important thing is, as we saw in the movie, the cell has to divide into two, right? Before dividing into two, what do you have to do? Each cell has to have exact DNA information, right? Before you decided to divide, you have to make exact one more copy of DNA, okay? That's very important in cell division, okay? That's what we go through most of it in this chapter. It's called mitosis, okay? So an introduction to cell division. So why do we need cell divide? Okay, some cells die each day, so you need more cells. Okay, you need to grow, you need more cells. The cell has to increase their number. Okay, there are times which needs quantity of new cells above, so you need more cells. So you have to generate some cells. How to generate? Before you divide, the first thing you have to think about is you have to make copy of DNA into DNA. We call it replication. In other words, duplication. You have to make exact copy of DNA. It's called replication. Okay. Second thing, physically you have to split those genetic information into two. Okay. It's called mitosis. The third stage is called cytokinesis. Cyto means cellular. Okay. So the rest of it, not the DNA, the rest of it will split into two. It's called cytokinesis. Okay? So the most important thing is division, mitosis. We'll go through in this chapter. But first, you have to make exact copy of DNA. Okay? Conceptually, how we can, so this is the whole thing, cell divide. Okay? So you start with one cell. You store DNA information into nucleus, okay? So cell decided to divide. Now I have to make 2 cells out of 1. Okay, what do? The first thing is you replicate DNA. You have to have 2 sets of DNA. Okay, and then split. Okay, finally separate. Okay, different thing is at some points you don't see the nuclear envelope. You don't see the separation between here and cytoplasm. Okay, so nuclear envelope, the membrane that wraps around DNA disappears and reappear. Okay, so this is the whole cell division. Okay, the first step, DNA replication. Okay, so the structure tells you how they can be exactly copied. Okay, so it's double helix, 2-strand DNA. Okay, you opens up, and each strand become template. Template means you can make copy of it. Okay, but you don't make exact copy of it. You put the complementary sequence in it. Okay, so by doing so, you can make new strand. Okay, based on this information. If this has G, A, T, C, you have to put C, you have to put T. It's automatic. Okay, that's how you make exact copy of it. Okay, yeah, this chapter is very, difficult chapter to follow. A lot of things coming on. Okay, but all of them are very interesting. Okay, because we can relate to. Okay, so but we saw the DNA replication, right? But DNA in our cell, okay, can have some different structures other than just double helix, okay? Because it's double helix, it's too long information. You have to put in small nucleus. What do you have to do? You have to somehow pack this DNA strand, okay? That's called chromosome, 염색체, all right? So DNA comes packaged in units called chromosome. Okay, so in other words, if you pull out from chromosome, and eventually it becomes double helix strand. Okay, but somehow you have to pack it, and you have to squeeze in to the nucleus. Okay, so in chromosome, not only DNA has it, there are some proteins involved in, but we don't have to worry about it at this point. Okay, so it's conceptually like this. Okay, starting with, we know that DNA has this double helix. Okay, and then you can have some protein called histone proteins. It's like beads on a string, your necklace. Okay, beads on a string. You wrapped around the string and the beads. Okay, and then think about traditional your home phone that has cord, right? Telephone cord that wraps around one more time and several more times. Now you start to see, you can see, not with your naked eye, but by microscope, you start to see. Okay, they are dense enough, you start to see. Okay, and those things are in nucleus. Okay, so so before cell divide, what do you have to do? You have to make exact copy of it. Okay, what do we call it? DNA duplication or replication would be a good one. Okay, DNA replication. Okay, if you figure it in chromosome, from here it becomes like this. Okay, you make one more identical strand of it. Okay, but it says unduplicated chromosome, not actual shape. What does that mean? Not actual shape. No idea? 한국말로.
복제되기 전에는 DNA가 이렇게 크로모솜 상태로 묻혀서.
Exactly. So you don't see this structure inside the cell. Why? Unless you decided to divide, there is no need to pack DNA like this. Okay? The only reason to pack like this is you're gonna move. Okay? you're going to move, you're going to split into two. That's why they pack the DNA. Okay? So in normal cells, you don't see this chromosome structure unless the cell decided to go cell division. They decided to go become two, then they start to pack. But when they pack, it has already been duplicated, already been replicated. Okay? So you don't see this structure inside the cell, but that's just conceptually, single chromosome should be look like this. But when we see, it's all already doubled. Okay? That's why we see only this chromosome, very short time. Okay? But what about the rest of the time inside the cell? It's like DNA strand is like spaghetti. It's all over. Okay? You don't pack that tight. You pack it, but it's loose enough you cannot see it. Okay? So here it comes. So this is chromosome number 1. Okay? One copy from your mother. Okay? This is also called human chromosome number 1. One copy from your father. Okay? So what do you have? You have 2 number 1 chromosome in your cell. Okay? So it says a paired chromosome. In other words, it's called homologous chromosome. In Korean, 상동 염색체. Okay? It's very similar, but not identical. Why? Your mother and your father doesn't have exact genetic information. They have different one. But why is it similar or identical? Chromosome number 1 has very similar structure. So if you say gene here determines your hair, it's just saying, right? Gene here determines your color of eye or something like that. The order of gene here located is very, very similar. Okay? That's why we call it homologous chromosome. 상동 염색체. Okay? So always chromosomes paired. So this is human chromosome called karyotype. Karyotype means, karyo means something about nucleus. So if you get your sample cells from your skin or whatever, and fix it with stain with something, at some point you can see this chromosome. Not always. Because these cells decided to divide, you catch it right before it. Then you can see this chromosome. Okay? So chromosome has been numbered by size. The biggest one is number 1. The smallest one is called 22. Okay? So from number 1 to 22, you have 2 chromosomes. So how many you have? 44. Okay? Plus X chromosome and Y chromosome. So this person is female or male? Male. Okay? Male has 1 X chromosome, 1 Y chromosome. Female has 2 X chromosomes. Okay? So this is human karyotype. Okay, if you have one more copy of chromosome 21, what do you have? You have Down syndrome. Okay, we'll come back to that. Okay? So now, so the cell decided to go cell division. Okay? Cell division is just like washing machine. Okay? Do you wash your clothes by yourself or your mother do the job? You run the washing machine. You put your clothes, dump your detergent, and then timer, you change it. Okay, what happened? The timer go, kick, stop, and go next step, wash, rinse, wash again. Exactly, cell cycle is the same thing. Okay, so cells start from here, okay, go through and divide. Okay, it's kind of growth. Okay, so they just divided. Okay, it start a new cell. We call it daughter cells. Okay, because it just, came out. And then they have to take some time to grow. Okay, So right after mitosis, the M phase is called G1. It's gap 1, the first gap. Okay. During gap 1, the cell become bigger, they make more proteins, they're ready for next division. Okay. Go to second phase is called S, stands for synthesis. They have to synthesize DNA. Why? They're replicating DNA. They make exact same copy of DNA. Okay, it's called S phase. And then they stop a little bit. Okay, it's called G2, gap 2. Okay, and then now they're ready to split. It's called mitosis. Okay, I know it's a lot of information. Keep coming, right? You have to be alert a little bit. Okay? Okay, questions? Nope? So what do you have to remember? Okay, what you have to remember is the cell goes around cycle. Okay, this is the beginning. Right after M, mitotic phase called M. Right after, there is a first gap called G1. And next step is cell decided to divide again. And then what do you have to do? You have to replicate DNA, right? It's called S phase. During this phase, they'll replicate DNA. Okay? And then they stop again, which is G2, and then they go through mitosis, okay, M phase. Okay. At the end, cytokinesis comes just automatic, okay? So G1 and G2. There can be also G0, which means they don't want to go through. They are done. They're done washing, okay? They stop. They don't divide anymore. Then it's called G0. Okay, so as long as you grow, you need more cell. The cell goes round and round and round. Okay, if your normal cell goes something wrong, and like this machine goes again, it becomes a cancer cell. They keep dividing. Okay, our cells says stop. You have to stop here, but cancer cells keep growing and growing. They dividing. Okay. So cell cycle is very, very important. Okay, so this G gap, G1, G2 is kind of checking point. Okay, everything is okay. If it is not okay, they stop there. But cancer cells, the mechanism is broken down. Okay. All right. So, wow, there's a lot of information. Mitosis and cytokinesis. So now we are focusing on mitosis, okay, M phase, okay. There are 4 phases coming, okay. You have to remember. I know it's biology. You have to remember, okay. The first, well, I'm telling you, easy way to remember. The first one is prophase, something before, okay. It's the beginning, prophase. The middle phase is called metaphase, okay. The third phase is anaphase, a little bit toward the end. Okay, the last step is called telophase. Telomere is the end of chromosome. Okay, it's the end of phase. Okay, what are the 4 stages? Prophase, metaphase, anaphase, telophase. Okay, what? Prophase, metaphase, anaphase, telophase. So now you have to see what happened, what's happening on each phase. Okay, so prophase. So now you start to see chromosome. Okay, before that it's like spaghetti. Okay, they've already been replicated. They've already been duplicated. Okay, now they are packing for moving. Okay, so they start to see. And there is a structure called spindle fibers and centrosomes. Yeah, okay. And then there is a nuclear envelope is breaking down. Okay, there is a nucleus is disappearing. Okay, that's the prophase. You're preparing for division. Second phase is called metaphase. Why? This one is easy. All the chromosomes are lined up in the middle. Okay, we call it metaplate or So in the middle, metaphase plate. Okay, so what happened in anaphase? We saw the movie, right? They start to pull from the one end. What is this end? Where did this end come from? It's coming from prophase, right? Okay. It's called centrosomes. Okay, centrosomes. Centrosome is they're the centered. They pulling chromosome into one side. Okay, it's called what is it called? Anaphase. Okay, telophase, everything is done. Okay, everything done. Okay, and then cytokinesis. You start with one cell, you end up with two cells. Okay, not that bad, right? No, it's bad. You have to remember all this. All right, so this is a picture of the last step, cytokinesis. It's like something is using belt squeezing two, okay? So in the animal cells, the cytokinesis happens from the outside and then shrinking and then dividing into two, okay? But the important thing is this one, okay? Prophase, what happened? Metaphase, what happened? Anaphase, telophase. Probably I'll give you chromosome condense, and nuclear envelope breaks down. What phase is it? It's prophase. Okay, all the chromosomes line up in the middle. It's metaphase. Okay, and chromosomes are pulled toward the centrosome, and then new nucleus are formed, which is anaphase, telophase. Sorry. Yeah. So in plant cell, it's a little bit different story because cytokinesis, you cannot squeeze the cell wall. So what do? You just put a new wall in the middle. Okay, plant cells. Okay, it's a little bit different. And what about bacterial cells? They don't have chromosome. Okay, they don't have nucleus, so it's a little more simpler. But usually their DNA is like rubber band. One single circular DNA is their chromosome. Okay, so you make copy and you split it into two. It's called binary fission. Okay.
The process of cell replication for some cells is fundamentally different from the mitosis and cytokinesis of eukaryotes. The cells of prokaryotes, bacteria, and archaea are an example. Bacteria are single-celled and possess a single circular chromosome. Bacteria cells do not hold their chromosomes inside a nucleus like eukaryotic cells. A bacterium has no nucleus. Instead, the chromosome of the bacteria cell is just attached to the cell membrane. Cell division for bacteria begins with the replication of the chromosome. The daughter chromosomes then attach themselves to different areas on the plasma membrane. The bacterial membrane and the cell wall pinch at a point between the daughter chromosomes, and the separation begins. The pinched ends separate the daughter chromosomes. When the ends close off, two cells exist where there was only one. This process is clearly quite different from what happens in eukaryotic mitosis and cytokinesis. Prokaryotic cell division is known as binary fission.
Okay, so now going back to chromosome story. Okay, so your cell has 46 chromosomes. Okay, so each chromosome are paired. In other words, if it is chromosome number 1, you have one copy from your mother, one copy is from your father. Okay, so we call it you have two sets of chromosome. Why? One set is provided by your mother, the other set is provided by your father. So we call it diploid, diploid. You have two sets of chromosome. Okay, so in normal cell division, think about what happened. Okay, so you don't see this chromosome because it's spread out already, spread out usually. Okay, they don't pack it unless they decide to move. Okay, whenever they decide to move and then pack, what happened? What you see is like this. You already copied the exact copy, right? So what do you call this each chromosome? It's called sister chromatid. Again, there are each sister chromatid. They are exactly identical. Why? They copied from the other. Okay? So it's a sister chromatid. I need one more hand though. Okay? Same thing. This is from your father. Okay? So during mitosis, what happened? Okay? So you start to see this duplicate chromosome at prophase. Okay? During metaphase, what happened? Okay, good. Thank you. During metaphase, what happened? They aligned in the middle of metaphase plates. Okay? Then what happened? They start to split in anaphase. In telophase, it's done. Okay? So what happened? They go like this. Right? You started one set of chromosomes, one copy from your mother, one copy from your father. Okay? At the end, you have exactly same set. Okay? So identical. Cell division, mitosis is making exact same cell. Got it? Right? So you have to understand this one because next one, meiosis, is a little bit different. We'll see the movie and then we'll have break. Okay?
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So next week is the midterm exam, okay? But I'm telling you, chapter 9 and 10 are more important than the other chapters, okay? I expect around 100 multiple choice questions, and so each chapter about 10 questions, but 9 and 10 can have more than 10, okay? So it's important to be awake, this one. So the second part, chapter 10, is about, we saw in the movie, right? It's about meiosis. It's preparing for the next generation. The key point of preparing next generation, preparing your sperm and your egg, is from 2 sets of chromosome, you have to reduce to 1 set of chromosome, right? The 1 set from your sperm and 1 set from your potential spouse egg or sperm, they'll meet, they'll become new individual with 2 sets of chromosome. Got the idea, right? We have to reduce into 1 set, okay? So what do you mean by from 2 sets to 1? This is, you have 2 sets of chromosome, okay? Either this one or this one. Okay, you're reducing into 1 set. We call it haploid. You start with diploid, okay? But end up haploid. That's your gametes. Gametes means sperm or egg, as we saw in the movie, okay? So how do you go through this meiosis? It's a little bit different from mitosis. So meiosis consists of 2 divisions. That's why they call it meiosis 1, meiosis 2, okay? Mitosis is just 1 single division, so they don't say 1 or 2, okay? So same thing, you start with already duplicated, okay? Already duplicated. What is this called? Sister chromatids, okay? Sister chromatids, they are identical, right? What do you call this one? A pair of chromosome, homologous chromosome, chromosome number 1. Okay, 1 from mother, 1 from father, okay? In mitosis, what happened? They replicated, they're independent, okay? They lined up in the metaphase plates, and then they'll split like this. Now you end up from your starting point, 2 sets of chromosome. This is mitosis, okay? Meiosis is different. How do they align? They aligned between homologous chromosomes. They align like this. 4 chromosomes align like this. It's called tetrad, okay? This is the prophase 1, okay? Sorry, prophase 1, they start to see, and they lined up like this, metaphase 1, okay? Sorry, I'll take it back, okay? Before aligned, okay? Sorry, they aligned, okay? They aligned like this, but still it's prophase 1, okay? One special thing happened in prophase 1, meiosis 1. What happened? They switch some part of homologous chromosome, okay? It's called homologous recombination, 재조합, okay? What does that mean? You're exchanging some genetic information between your mother's chromosome and father's chromosome, okay? They are homologous chromosome. They're supposed to be very similar, but not identical, because your mother and your father is different, okay? But when you were born, you got mixed up, okay? So you don't have to just like your mother. You don't have to just like your father because it's been mixed. Okay? When this thing happened, prophase 1. It's in meiosis, only meiosis. Okay? And then second, metaphase 1. They all aligned up. This is chromosome 1, this is chromosome 2, this is chromosome 3, this is chromosome 4. Okay? Now you have one more choice. Okay, one more choice. What do you mean? Your chromosome number 1 can align this way or this way. What does it make any difference? If you go split this way, this will have mother's copy, right? But if you align this way, this will have father's copy. You have choice because that's the meiosis. You start with this. Your choice is either you will take this one or you will take this one. Okay, you have choice. Okay? Too complicated? Okay, one more time. Going back. So this is mitosis. Okay, the purpose of mitosis is you're generating exactly same cell. Okay, from diploid to diploid, which means you have 2 sets of chromosome. You will have 2 sets of chromosome. Okay, you have 2 sets of chromosome. Okay, you go through cell cycle and go through G1, S. You duplicate it. You're now at the G2 and you're at the mitosis M. Okay, so what do? You align like this. Okay, align between sister chromatids. Even if you change something, it doesn't matter because they're exactly same. You just copied it. Okay, so there's no exchange. What do? You just simply split like this. Okay, so you start with diploid, end up with diploid. That's mitosis. Now, meiosis is a little bit different. Okay, the purpose of meiosis is from 2 sets of chromosome, you either one set of it, either this one or this one. Okay, that's your gamete, sperm or eggs. Okay, so in meiosis 1, prophase 1, okay, what happened is alignment between homologous chromosomes. Homologous chromosomes. Okay, that's different from mitosis. And also, some part of chromosome is exchanged. It's called homologous recombination. Okay? And then what happened? Metaphase that align chromosome 1, chromosome 2, chromosome 3, chromosome 4, and anaphase 1, what happened? Split like this. Okay? Split between homologous chromosomes. This is meiosis 1. At the end of meiosis 1, is it haploid? Is it diploid? You have 2, but you have 1 set. Okay, you have 1 set of chromosome. That's haploid, 1 kind. Okay, that's meiosis 1. Meiosis 2 is simple. Just split between these 2. Okay, meiosis 2 is just split into 2. At the end, what do you have? Haploid. Okay, all right. At the end of your sperm, an egg has 1 set of chromosome 1. Okay, all right. Question? Clear? All right. So the meiosis is preparing for sexual reproduction. In other words, why we have to reduce chromosome set? Because sperm will meet egg eventually. Okay, you will get married, you will have your baby. That's why. Okay, an overview of meiosis. Okay, all the human cells are diploid except your germ cells, cells preparing for the next cell, next generation, egg or sperm. Okay, they are haploid, a single set of chromosome. Okay, so haploid cells are produced through meiosis, called gametes. Okay, gametes means egg or sperm. Sperm means singular, both plural. Okay, 단수 복수 똑같은 단어예요. sperm. Okay, so these are called reproductive cells, 생식세포, or germ cells, whatever you call it. Okay, so now compared to mitosis. Meiosis consists of two divisions, meiosis 1 and meiosis 2. Okay, prophase 1, which means meiosis 1, the first step. Okay, you cannot draw this thing, that's why you have to be careful. Okay, they align between homologous chromosomes, red and black. Okay, then some is exchanging. Okay, it's called homologous recombination. Okay, and then aligned up like this. Okay, and then split. Okay, that's meiosis 1. Meiosis 2 is very simple because they are splitting between sister chromatids. They're identical one, except some of them are exchanged. Okay, that's meiosis. So you go through all of them, try to remember. Okay, and so important stage. Okay, important stage is this prophase 1. What happened? Homologous recombination happens, right? Second stage, metaphase 1. What do you have? You have choice of either line up this way or this way. That will make difference in terms of genetic information. Okay, that will make difference which sperm will have which chromosome eventually, or which egg has which chromosome. That will be diversity of your children. Your children look like you, but they are not you. Okay, they don't think like you. They're totally different. Okay. All right, so eventually, if you get married, have sex, what happened? Sperm meets egg, and then fertilized egg. It's called zygote. It's your baby. Okay, starting. Okay, so steps in meiosis. Meiosis has two divisions: meiosis 1, meiosis 2. Okay, so important thing happened in prophase 1. Okay, prophase 1. It's called exchange reciprocal section. What do you mean? Same part is exchanged. It's explaining homologous recombination. Okay. So more elaborate pictures. The prophase 1, the homologous chromosomes aligned. They exchange some part of it. Okay, it's called homologous recombination. Metaphase 1, align between homologous chromosomes. Okay, separate in anaphase and telophase 1 and meiosis 2, prophase 2, metaphase 2, anaphase 2. So if you see prophase 1, metaphase 1, it means those are meiosis, right? Just metaphase, anaphase, it's mitosis. Okay, don't get confused. So why you go on through this meiosis? What is the significance of sexual reproduction? Okay. So meiosis generate diversity. What do you mean diversity? So human chromosome has two sets. We have 46 chromosomes and two sets. One set from your father, one set from your mother. Okay, so theoretically, you have choice. Number one, chromosome, either you can pick your mother, either you can pick your father. Chromosome number 2, the same thing, right? You can, two choices. So how many choices you have to make one sperm? You can have 23 chromosomes, right? You can pick, so 2 times, not times, it's what do you call it? 2 over 23, that combination, okay? So even if your children is totally different. Okay? So you have choice. Plus, what do you have? Crossing over. You can exchange some part of it chromosome. There are two steps you can have diversity. Okay? Meiosis, unlike regular cell division or mitosis, which produce daughter cells that are exactly genetic copies of, this is mitosis, right? Meiosis generate genetic diversity in two ways. What are those two ways? First, this is important. In prophase 1, meiosis, homologous chromosome pair with each other and called crossing over, recombination, homologous recombination, or crossing over. They change some part of it. Okay, that's number one. What is number two? Metaphase 1 depends on how you align chromosome. You can choose either side, mother side or father side. That's the second event. Okay? So, so animal evolved, okay, so that sexual production is dominant these days. Male, female, okay, they'll make gametes, they'll make reduce half of the one set of chromosome. Why? Why is it? This will generate diversity in progenies. Your next generation will different from you. Okay? The data says female students have much higher chance of finishing college in 4 years. Remember, male students in the United States, they don't have to go to military, but they cannot just finish college in 4 years. I don't know what they're doing, but they can finish. Okay, second statistics. In over 1,000 cities in the United States, 1,000 cities, it's a small, big, middle size, all the cities. They calculated employments. Okay, for the first time in US, female employment surpassed male employment. Although there's still salary gap, but employment itself, female surpassed male. So what's the point? In the United States, it used to be, if you're a male, okay, if you're just graduate high school, okay, you don't have specific education, but you still can get a good job. In the Midwest, for example, car company and a manufacturing company, you get a job. It's job for life. It used to be. We're next, previous generation. It's a lifelong job. It guarantees you middle class salary. So male you don't have to do anything, okay? But now, economy in US has changed. There is no such manufacturing jobs anymore. So you have to thrive for higher education. The males just can't do it. Females are doing it better in terms of. So it will come at some point in Korea, not in this generation, but perhaps next generation. Okay? So if your parents invest to your daughter, not son. Okay? But my generation, I'm the youngest out of 7 children. Okay? So if you're parents, you have 7 children, you don't have enough money. Who do you send to college? Who do you want to send? First son. That was then, one generation before. Okay, that's the highest chance he will get a job. He will support the family, right? Now, it's different. Okay, so read that book. It's an interesting one. Okay, so how do you make egg or sperm? Okay, it's a little bit different. Okay, so let's go through. Yeah, so how do you determine the sex, male and female? Okay, so mother has 2 X chromosomes. Okay, and go through meiosis. Okay, if you go through meiosis, it will divide it twice. Okay, and then what happens is the first meiosis is split into 2. Okay? Although this looks same, but they are not same. They are homologous chromosome. One X coming from father, one X coming from mother. So it's different. Okay? But they will split between homologous chromosome in first meiosis. Now they become haploid. Okay? Second chromosome is just division between sister chromatid. They are identical ones. Okay? So you can generate 4 gametes. Okay? Male, same thing. Okay? The first split between X and Y, and then split between sister chromatids. Okay? So each one is one sperm, each one is one egg. So how do you produce female? XX meets female, XY meets it's male. Okay? So who determines? So the sperm will determine whether it's a boy or girl. Okay? So I told you mitochondria has some DNA, right? All of your mitochondria is coming from your mother. Why? The sperm doesn't take any cytosol. They just take the DNA from the nucleus. They just put the DNA into egg. All the cytosol, cytoplasm is coming from your mother. Okay? So if you sequence mitochondria DNA, you can follow your mother's side history. Okay? All right. Gamete formation in humans. Okay, so gamete formation means how to make sperm, how to make egg. It's very complicated. So a few more words. Okay, spermatogonia, primary spermatocyte, secondary spermatocyte, spermatids, sperm. What do you do? Okay, you have to remember. Okay, so I'll not ask tough questions, but there is a difference, big difference. Okay, I'll tell you what's the difference. Sperm. This is how to make sperm. Okay, you start with gonia, gonium, spermatogenesis. Oogenesis means you're making something. Genesis, 창세기도 되고 차 이름도 되고. Okay, Genesis, you're making something. Okay, sperm making, spermatogenesis, egg making, oogenesis. Okay, starting point, spermatogonium. Oogonium is the original 원조. Okay, and then primary spermatocyte, primary oocyte. Okay, the difference is when they split, primary spermatocyte, primary spermatocyte will generate one more spermatogonium. Okay, so what I'm trying to say is sperm has stem cell-like character. Stem cells, if you divide into 2 cells, one cell contains the characteristics of your parent cell. Okay, so in other words, sperm is you keep one cell as a stem cell, okay, and then split one primary spermatocyte, go through meiosis and generate 4 sperm, okay, and still keep this stem cells, which means once you start to make sperm, when do you start to make sperm? You become adolescent, puberty, 사춘기. Okay, you start making sperm. Okay, and then when do they end? Until the day that you die. In male case, they will keep generating sperm. That's why Picasso had their son or daughter when he was 80 something. Okay, they'll keep produce this sperm. Okay, but female is different. Okay, once they generate primary oocyte, okay, they have limited number early in your baby. Okay, and as you go up, it will stop at some point. Okay, and then you start again this meiosis when when you start a cycle. In other words, when you become adolescents, okay? And each month you use one egg. And how many eggs eventually you will use? Let's say from 10 to 50, 40 years, 12 months, 500 is the maximum. 500 eggs, that's it, no more. Okay, when you go through menopause, your mother probably go through menopause, no more eggs produced. Okay, so the strategy of having more children is quite different from male and female. Okay, female, you have only limited number of eggs. What do you have to be? You have to be selective. You don't just date and then get married. You have to be very, very selective. Okay? On the other hand, male case, you're producing your sperm forever. So your strategies go through, so we'll see. Okay? So that's different. So go through it. All right. Asexual reproduction means you don't go male, female. Okay, you just make identical progenies. Okay. For example, bacteria, we saw it from one, it's split into two identical ones. You don't have to make it different one, but it's a lot more efficient than sexual reproduction. But sexual reproduction is more generating diversity. Okay, that's the whole point. And then there's some vegetative reproduction. Vegetative reproduction means a lot of cases in plants. Okay, if you cut the plants and plant it, they'll grow. Okay, that's the vegetative reproduction. Okay, so they are clones, genetically identical. You cut, you put it, they grow. Right, that's the vegetative. And some animals regenerate. Okay, so that's it for chapter 10. I'll post for the edition each chapter summary question, right? I will post the answer to the online class. I'll not go through all the reviews today, okay? But each chapter you have to read and then solve the questions. 아직 중간고사 공부를 안 했으면 10장서부터 시작하죠. 10장서부터 거꾸로 내려가죠. 오케이? 10장, 9장, 8장. 오케이? 쭉 한번 읽는데 영어책이 너무 많이 잘 안 읽힌다. 뒷부분에 서머리 부분이라도 읽어. 오케이? 그런 다음에 연습 문제 풀고 앞장으로 넘어가고. So you have to read, if you cannot read the whole thing, read the chapter summary and solve the questions. And I'll see you next Tuesday, 11. Okay? We're going to have midterm exam. Okay? 질문 있으면 온라인 강좌로 이렇게 올리든지 그럼 내가 답을 해줄게. Okay? Right. Good luck.